Okay, in this session we're going to cover some basic bone physiology. We're going to look at the different cell types that form bone and regulate bone turnover, and we're going to look at how defects in these cell types, which are the osteoblasts which make bone and osteoclasts that resorb or break down bone, we're going to look at how defects in these might cause specific bone diseases. And then we'll move on to a couple of bone diseases, Paget's disease and osteoporosis, and we'll look at some pharmacological treatments that target either osteoblast or osteoclast activity, mostly osteoclast activity, to try to treat these diseases. So the main point of this slide is to let you know that bone's a dynamic tissue in that it is remodeled over time. So we're looking at about 10% of the entire skeleton being remodeled every year, and this is in response to bone flexing, micro cracks, micro stresses on the bone when you allow impacts to occur. So this accounts for the 10% of turnover, and if you do not do any bone flexing, the net result is bone loss, because the way bones remodel is that where stronger bone is needed, where they're flexing more, then they're built up to strengthen them to reduce excessive bone flexing just from normal day-to-day activities. So immobility, being paralysed, being weightless and so on will result in a reduction in bone mass because there's no signals telling the bone to be built up and strengthened where required. If we get an excessive amount of generalised bone loss, so if bone degradation exceeds bone formation, we're talking about diseases such as osteoporosis, so typically more associated with postmenopausal women, although not entirely, and then bone formation excesses, many congenital diseases result in excessive bone formation, which we term osteopetrosis. So this can be where the cortical bone is so thick that that most of the bone marrow cavity is contained by bone. So just a quick overview of the bone structure. If we're looking at a typical long bone here, we have got at the ends of the bone we have this cancellous or trabecular bone, also sometimes called honeycomb bone, and this is very strong and very lightweight. We have cortical bone, which is the solid, what you traditionally see as solid bone. That's called cortical bone. Within the centre we have the bone marrow cavity, and within the bone marrow cavity and associated with the trabecular bone is where we get the bone marrow stem cells, hemopoietic stem cells, mesenchymal stem cells, which produce all of the white blood cells, and mesenchymal stem cells go on to produce the precursors of connective tissue cells. So this is a cross section through some bone, and this is taken microscopically. So we've taken some trabecular or honeycomb or cancellous bone, and we have got a very thin, maybe 5 micrometer section through it that's been sliced on a microtome. So this is how a pathology specimen would be looked at. We'd take a section of bone, slice it very thin, put it on a microscope slide, and then stain it up with some stains to see what cells are in there. So all of this region here is the bone. So this is collagen. It's been stained up with a particular stain that just stains mineralized collagen. Embedded within those are osteocytes. We'll come back to them later. This large cell is an osteoclast. It's a white blood cell derived cell, and it's a multinucleate, so it's got several nuclei because it's several cells fused together. There's another one, and its job is to eat away at bone where bone remodeling is required. We've got the osteoblasts. These synthesize new bone, so these are derived from the mesenchymal stem cells, which are the precursors to all connective tissue. And what you can see here is where they've got this nice pinky purple stain, there's a halo of unstained bone, and that's where we've got unmineralized collagen. And collagen is the protein that these osteoblasts secrete, and it's laid down and then mineralized, and that is what becomes hard bone. Now the osteocytes are cells encased within the collagen, and they link together as a network of almost neuronal-like cells in the way that they communicate with each other, but they're not neurons. And these are basically osteoblasts that have been abandoned in the bone matrix, and these are the cells that are going to sense cracks. So if a crack appears across this bit of bone, these cells here will sense it and pass on a signal to the osteoclast to come and eat away at the bone and clear up any debris, and then osteoblasts to come in and secrete new bone. So, as I sort of alluded to earlier, we've got we have a normal genetic baseline of bone mineral density, and by doing exercise and mechanical loading we can increase the bone mass. And if we don't do any exercise, for example, if think of a paralyzed patient or an astronaut, for that period of time they will lose bone mass, because we only build up new bone when it's required to strengthen a weakened bit of bone. So if you click on some of these links here, these will take you to some little animations that just show this process going on. So osteoporosis is a disease of bone where we tend to get reduced bone mass, and this is often most obvious in the trabecular bone at the ends of the long bones. So this is showing normal bone with very tight knit of meshwork of like a honeycomb structure, whereas in osteoporosis we've got these much more open circles where there's no bone material, and across this section this will be much weaker and much more prone to fractures, which is why when you have postmenopausal elderly ladies, first sign of winter, they slip on the ice, fall down, fracture the hip, and it's partly because of this reduced trabecular bone at the head of the femur, which is much weaker, so as soon as they hit the ground they're much more likely to fracture. And that is often the first sign of osteoporosis in some patients, is that they fall, they fracture, they have a bone scan and find out they've got reduced bone mass. So osteoporosis, it can be due to excessive osteoclast activity or diminished osteoblast activity, and with most people thinking it's the diminished osteoblast activity that is the problem. So we've got osteoclasts working at the normal rate, remodeling bone, osteoblasts trying to fill in the holes that are being made by the osteoclasts and not completely filling in those holes, resulting in a net bone loss. So it's a disease that affects postmenopausal women are the major group who are affected, and this can be related down to hormonal changes during the menopause. So during the menopause, women can lose up to 20% of their bone mineral density. If this continues untreated, you can then get osteoporosis combined with factors such as a poor diet and relatively little exercise and a sedentary lifestyle. So, and we'll come on to the hormonal reasons shortly. So this is just showing the sorts of fractures that occur in osteoporosis. So we've got an osteoporotic vertebral compression fracture there, where the bone has fractured and then become compressed, so that each vertebra is smaller, as a smaller depth than a normal vertebra, and this is a fairly common sign of osteoporosis. So we can measure the degree of damage in osteoporosis by doing a bone mineral density scan, or a DEXA scan, dual X-ray absorptimetry test. So this is just looking at doing an X-ray of the bone and looking to see what the bone mineral density is. And if we think of everyone's bone mineral density being on a normal distribution, So think of a normal distribution with standard deviations containing one standard deviation from the mean, plus or minus that, containing about 60 odd percent, and two standard deviations, 90 odd percent of patients. Patients who are more than 2.5 standard deviations below the normal range are classed as osteoporosis. So this is just a scan that this is important when you're looking at patient notes, because you may well see a T-score on the notes. So if the T-score is less than 2.5, that means the patient has got osteoporosis, whereas -1 to 2.5, -1 to -2.5, we're talking about osteopenia, generalized low bone mineral density, but not classed as osteoporosis yet. And these are patients that you may want to have an intervention to prevent excessive bone loss. These are patients who need therapy for their excessive bone loss. So, looking at the changes in bone mineral density with age, once you get beyond 40, both men and women lose bone mineral density, but it's much more marked in women post-menopause or during the menopause. And this is thought to be down to estrogen levels in particular. And most bone biologists think that it's estrogens, high levels of estrogens maintain the osteoblast lifespan, whereas in lower levels of estrogens, the osteoblasts will differentiate, so they'll be produced from mesenchymal stem cells, and then they will undergo something called programmed cell death or apoptosis. So if you can imagine a 40-year-old woman, their osteoblasts might hang around filling in holes for, I don't know, a month or so, whereas someone who's postmenopausal, those osteoblasts might hang around for maybe a couple of weeks and not completely fill in the resorption pit that has been created by the osteoclast. So if the osteoblasts don't hang around and do their job for as long, then there's going to be a generalized bone loss over time. So the evidence for a steroidal hormone link is quite strong. Both testosterone and estrogens affect osteoblast survival. Now we can look at treating osteoporosis later when we look at bisphosphonates, which counterintuitively are osteoclast targeting drugs. So before we move on to bisphosphonates, we'll look at Paget's disease. And this is caused by a local overproduction of bone. So this is overproduction of bone due to osteoblasts overgrowing. And overgrowing having excessive activity and also refusing to die when they're supposed to. So this is called Paget's disease. We have pagetoid bone, which is newly synthesized bone, which is susceptible to fractures, and we tend to get this asymmetrical distribution of localized regions of bone. So this is a different bone scan. This will be a technetium-99 bone scan, which is a radioactive isotope that accumulates where new bone is being produced. So here, all through the pelvis, this hip, this tibia, are all producing excessive amounts of bone compared to the normal skeleton. And as you can imagine, this is very painful. And there's another case looking very similar. So we know this is caused by uncoupled osteoblast and osteoclast activity. We think it's most likely due to excessive bone formation due to the osteoblasts being unable to regulate their growth. And there's some strong evidence for this in that Paget's disease patients sometimes go on to develop osteosarcoma, which is a malignant tumour of osteoblasts. So that's suggesting that this might represent a dysregulation of osteoblasts, and then if those cells develop additional mutations later on in life, they can develop on into osteosarcoma in a very small proportion of patients. So this is a fascinating disease to study from that respect, because it may give us the sort of the early changes that are happening prior to osteosarcoma starting, although the vast majority of patients do not get osteosarcoma. Okay, so that's part one. I'll start part two in a moment.